Today, we're diving into the fascinating world of cranial nerves, drawing insights from a comprehensive lecture presentation by Professor Vanessa. Cranial nerves are a vital part of our peripheral nervous system, directly connecting to the brain. They're classified in a few key ways. Some are primarily sensory, carrying somatic sensory information like touch, pressure, vibration, temperature, and pain. Others are special sensory, handling information for our unique senses, such as smell, sight, hearing, and balance. Then we have motor nerves, which transmit somatic motor commands, and finally, mixed nerves, containing both sensory and motor fibers. Let's begin our journey with the first cranial nerve, the olfactory nerves. Their primary function is special sensory, specifically for our sense of smell. These nerves originate from the receptors of the olfactory epithelium in the nasal cavity. They pass through the olfactory foramina within the cribriform plate of the ethmoid bone. Their destination is the olfactory bulbs, which are neural masses situated on both sides of the crista galli. Here, the axons forming the olfactory nerves synapse with postsynaptic neurons. The axons of these postsynaptic neurons then go on to form the olfactory tracts, transmitting the smell information further into the brain. Next, we have the optic nerves, cranial nerve 2, responsible for special sensory vision. They originate from the retina of the eye and pass through the optic canals located in the sphenoid bone. Their destination is the diencephalon, reached via the optic chiasm. This chiasm is a crucial point where fibers from the medial half of each retina cross over to the opposite side of the brain. After this crossing, the reorganized axons continue their journey to the lateral geniculate body of the thalamus, forming what are known as the optic tracts. Moving on to cranial nerve 3, the oculomotor nerves. These are motor nerves primarily controlling eye movement. They originate in the midbrain and pass through the superior orbital fissures of the sphenoid bone. Their destination is twofold. They provide somatic motor control to several eye muscles, including the superior, inferior, and medial rectus muscles, the inferior oblique, and the levator palpebrae superioris. They also provide visceral motor innervation to the intrinsic eye muscles, which is achieved by innervating neurons of the ciliary ganglion. Cranial nerve 4 is the trochlear nerves. Like the oculomotor nerves, their primary function is motor control for eye movement. They also originate in the midbrain and pass through the superior orbital fissures of the sphenoid bone. Their specific destination is the superior oblique muscle, helping to control its movement. Then we have the abducens nerves, cranial nerve 6. These are another set of motor nerves dedicated to eye movement. They originate from the pons and, similar to the oculomotor and trochlear nerves, pass through the superior orbital fissures of the sphenoid bone. Their destination is the lateral rectus muscle, responsible for abducting the eye or moving it outward. Let's turn our attention to the trigeminal nerves, cranial nerve 5, which are quite complex. They're mixed nerves, providing both sensory and motor functions to the face, and are actually the largest of the cranial nerves, featuring 3 major divisions. The ophthalmic nerve handles sensory input for orbital structures, the cornea, nasal cavity, and skin of the forehead, upper eyelid, eyebrow, and part of the nose. The maxillary nerve provides sensation for the lower eyelid, upper lip, gums, teeth, cheek, nose, palate, and part of the pharynx. The mandibular nerve is responsible for sensory information from the lower gums, teeth, lips, palate, and part of the tongue, and it also contains motor fibers originating from the motor nuclei of the pons. These divisions pass through specific openings, the ophthalmic nerve through the superior orbital fissure, the maxillary nerve through the foramen rotundum, and the mandibular nerve through the foramen ovale. Their sensory fibers travel to the sensory nuclei in the pons, while the motor fibers of the mandibular nerve go to the muscles of mastication, essential for chewing. A notable structure associated with these nerves is the semilunar ganglion, which contains the cell bodies of the sensory neurons. A painful condition known as tic douloureux or trigeminal neuralgia can affect the area innervated by the maxillary and the mandibular nerves. Moving on to the facial nerves, cranial nerve 7, another mixed nerve involved in both sensory and motor functions for the face. Its sensory origin is the taste receptors on the anterior 2/3 of the tongue, while its motor component originates from motor nuclei in the pons. These nerves pass through the internal acoustic meatus, leading to canals that end at the stylomastoid foramina. Their sensory destination is the sensory nuclei of the pons. For motor functions, they provide somatic motor control to the muscles of facial expression and visceral motor innervation to the lacrimal gland, nasal mucus glands, and the submandibular and sublingual salivary glands. The facial nerve also branches into 5 main parts: temporal, zygomatic, buccal, marginal mandibular, and cervical. Specific structures include the geniculate ganglia, containing sensory neuron cell bodies, and postganglionic fibers from the pterygopalatine ganglia, which innervate the lacrimal glands and glands of the nasal cavity and pharynx. Postganglionic fibers from the submandibular ganglia innervate the submandibular and sublingual glands. A common disorder called Bell's palsy, often due to viral infection causing inflammation of the facial nerve, leads to paralysis of facial muscles on the affected side and a loss of taste sensation from the anterior 2/3 of the tongue. Cranial nerve 8, the vestibular-cochlear nerves, are special sensory nerves. They consist of 2 main parts: the vestibular nerve, essential for balance and equilibrium, and the cochlear nerve, dedicated to hearing. Both originate from receptors in the internal ear and pass through the internal acoustic meatus of the temporal bones. Their destination is the vestibular and cochlear nuclei within the pons and medulla oblongata, respectively. The vestibular nerve originates at the receptors of the vestibule, which is involved in balance, and connects to the vestibular nuclei. The cochlear nerve monitors receptors in the cochlea, which is responsible for hearing, and connects with the cochlear nuclei, both within the pons and medulla oblongata. Next, we have the glossopharyngeal nerves, cranial nerve 9. These are mixed nerves providing sensory and motor functions to the head and neck. Their sensory origins include the posterior 1/3 of the tongue, parts of the pharynx and palate, and the carotid arteries. The motor component originates from motor nuclei of the medulla oblongata. These nerves pass through the jugular foramina, situated between the occipital and temporal bones. Their sensory destination is the sensory nuclei of the medulla oblongata. For motor functions, they provide somatic motor control to the pharyngeal muscles involved in swallowing and visceral motor innervation to the parotid salivary gland. Associated structures include the superior and inferior ganglia, which house sensory neurons, and the otic ganglion, where visceral motor fibers synapse. Cranial nerve 10, the vagus nerves, are also mixed nerves, but uniquely, they are widely distributed throughout the thorax and abdomen. Their sensory origins include parts of the pharynx, the auricle and external acoustic meatus, the diaphragm, and visceral organs in both the thoracic and abdominal pelvic cavities. The motor component originates from motor nuclei in the medulla oblongata. These nerves also pass through the jugular foramina. Their sensory destination is the sensory nuclei and autonomic centers of the medulla oblongata. For visceral motor functions, they innervate muscles of the palate and pharynx, as well as muscles of the digestive, respiratory, and cardiovascular systems found in the thoracic and abdominal pelvic cavities. Like the glossopharyngeal nerves, the vagus nerves have superior and inferior ganglia that contain sensory neurons. Penultimately, we have the accessory nerves, cranial nerve 11. These are motor nerves, primarily controlling muscles of the neck and upper back. They originate from motor nuclei in both the spinal cord and the medulla oblongata, and they pass through the jugular foramina. Their destination depends on the branch. An internal branch goes to voluntary muscles of the palate, pharynx, and larynx, while an external branch innervates the sternocleidoid and trapezius muscles. The accessory nerve structures include a spinal root, which contains motor fibers originating in the anterior gray horns of the first 5 cervical segments of the spinal cord, and a cranial root, with motor fibers from the medulla oblongata. The internal branch joins the vagus nerve to innervate swallowing muscles and vocal cord muscles, while the external branch directly controls the sternocleidoid and trapezius muscles. Finally, cranial nerve 12, the hypoglossal nerves. These are motor nerves specifically for tongue movements. They originate from motor nuclei of the medulla oblongata and pass through the hypoglossal canals of the occipital bone. Their destination is directly to the muscles of the tongue, enabling speech and swallowing. Beyond individual nerve functions, it's also important to understand cranial reflexes. These are automatic responses to stimuli that involve the sensory and motor fibers of cranial nerves. They can either be monosynaptic or polysynaptic reflexes and are clinically useful for checking the condition of specific cranial nerves and various parts of the brain. This detailed overview of the cranial nerves and their functions, as presented by Professor Vanessa, highlights their critical role in governing essential sensory and motor processes throughout our head, neck, and even extending into the torso. It's a testament to the intricate design of our nervous system. Consider sharing this episode with someone curious about how our brain controls so many vital functions.